Besides water, proteins are the most abundant molecules in all known forms of life. 70% of your body weight is water, and about 17% is protein. Proteins are the most diverse class of biological molecules, making up everything from enzymes and hormones to antibodies, and muscle is full of them. In this lecture, we'll learn about the 20 kinds of amino acids strung together in proteins. Because amino acids are the building blocks of all proteins, all cells need amino acids. We see amino acids in shampoos, but not on food labels. How can something so seemingly important be almost invisible from a nutritional standpoint? Should you take amino acid supplements? Can they help you get to sleep, prevent or cause eruptions of herpes virus, slow cancer? We'll touch on these questions and more in this lecture on amino acids. Amino acids found in proteins all have the same simple core structure: a central carbon, an amino group with an NH2, and an acid group with COOH, as well as a lone hydrogen opposite the R group. Change to a bond line drawing, and this is the same thing. We just don't write out the carbons. The central carbon is the alpha carbon. Attached to it is an amino group, a carboxyl group, a hydrogen, and a fourth group that varies in structure between amino acids called an R group. For this lecture, we're going to focus on the R group, where R is the organic chemist's designation for radical. We'll use it to mean remainder. The R group is the only part of an amino acid structure that varies from one to the other. The other parts of the structure are common to all of them. Now, R can be just one additional carbon plus 3 more hydrogens. That makes alanine. Or 3 carbons in a V shape plus some more hydrogens, making valine. We refer to the R groups as being aliphatic when they contain only carbons and hydrogens, like these amino acids I just mentioned. Want to know the properties of a specific amino acid? It's simple. Look at the R group. Aliphatics have only carbons and hydrogens, which are so similar in electronegativity that they are nonpolar, meaning that they are hydrophobic. They can't make hydrogen bonds with water, and so they avoid it. Other R groups contain other atoms and can ionize or make hydrogen bonds, so these are the hydrophilic amino acids. They like water. About half of the amino acids are nonpolar and hydrophobic. These are the inward-looking introverts of a folded protein molecule. The other half are polar, water-loving extroverts. You gotta have both. Cells are really choosy. Over 300 amino acids exist in nature, but only 20 are coded for in our DNA. Amino acids, of course, are the building blocks of proteins. The bonds holding proteins together are known as peptide bonds, and they form by joining the carboxyl group of one amino acid to the amino group of the next. All amino acids except glycine can exist in 2 mirror image forms. These differ in the arrangements of the 4 groups around the alpha carbon. It's like left and right hands that cannot be superimposed on each other. Here's alanine, the simplest chiral amino acid, in both forms. The 2 forms are called stereoisomers. One is the L form, the other is the D form. Glycine is different from all the other amino acids in having an H across from another H. So there's only one form of glycine. Molecules with handedness are called chiral. Why does this matter? Well, it turns out that all chiral compounds, like amino acids, when made apart from cells, have a 50-50 mixture of the D and L forms. However, amino acids made in cells for use in protein synthesis are almost completely in the L form. This is because the enzymes synthesizing these amino acids have specific 3D structures, which arrange reactants such that only one form can be made. We can tell if a mixture of molecules was made by an enzyme or by a non-biological chemistry simply by analyzing its chirality. If non-enzyme chemistry produced the molecules, they'll have equal amounts of D and L forms, since that's what ordinary chemical reactions in test tubes yield. Molecules made by enzymes, though, will have one form or the other. In cells, the enzymes almost always make the L form. Only very rare amino acids are made by cells in D configurations. The stereochemical bias of amino acids made by enzymes is also used by scientists trying to detect signs of extraterrestrial life. It is not uncommon, for example, for meteorites to contain amino acids. A strong bias for either of the two forms would suggest a much higher probability that they were made by a living organism than a 50-50 mix of DNL. Let's now look more closely at the 20 amino acids used in building proteins, starting with amino acids whose R groups are hydrophobic. Hydrophobic amino acids, as noted, have R groups mostly containing carbons and hydrogens. This includes glycine, alanine, valine, leucine, and isoleucine. The degree of hydrophobicity increases steadily from glycine to isoleucine as the R groups increase in size and complexity. Glycine, we've seen, is the simplest amino acid with a lone hydrogen as its R group. Despite its simplicity, glycine has important functions in proteins, especially in collagen, the most abundant protein in the human body. The small size of glycine's R group is actually a useful feature as it allows it to function in places where an amino acid with a larger R group could not. Collagen is a tightly wound triple helix molecule, and it's the presence of tiny glycine located at every third amino acid of each helix that allows collagen helices to wind so tightly. Glycine also acts as a signaling molecule for transmitting nerve signals in the spinal cord, brainstem, and retina. One of the mechanisms of action of the poison strychnine is blocking or impairing the action of glycine in nerve transmission proteins. The second simplest amino acid is alanine, having a CH3 methyl group for its R group. It is one of the amino acids sometimes seen in the D form in cells. One place we find D-alanine is in the cell wall of bacteria, where it is linked to other amino acids to form supports for the cell wall. The integrity of the cell wall is important for all cells. Mess with it and a cell dies. Penicillin, for example, kills bacteria by interfering with their ability to make cell walls. So bacteria are pretty protective about their cell walls, and using D-alanine is a strategy for protecting them. Since D-alanine is not a component of normal proteins, enzymes that would otherwise attack and destroy amino acid chains in cell walls can't touch the ones that contain D-alanine. The amino acids valine, leucine, and isoleucine are sometimes referred to as the branched chain amino acids, or BCAAs. These amino acids have R groups with longer chains of carbon and hydrogens, making them more hydrophobic than glycine or alanine, because hydrophobic properties are somewhat additive in nature. Longer means more. Like the other amino acids, they are important for making proteins. Unfortunately, there is a widespread belief that BCAAs are especially effective in helping build muscle. This leads some people to load up on BCAA supplements. That could actually be doing harm because BCAAs are present at higher levels in the blood of people with insulin resistance associated with type 2 diabetes. Worse, recent studies since 2015 suggest that high levels of BCAAs in the blood actually cause insulin resistance. So it would seem that getting too much of valine, leucine, and isoleucine is not a good idea. In fact, experiments in mice have suggested that a diet low in BCAAs promoted leanness and improved control of blood sugar. Two other amino acids, proline and methionine, sometimes get grouped with the hydrophobic amino acids we've been discussing. Both are oddballs and share characteristics with the nonpolar amino acids. Proline has a formula almost like valine, but instead of making a V, its R group loops around from the alpha carbon to bond to the alpha amine group, almost like proline is trying to make a perverse letter P. The resulting ring structure is unique among the amino acids because it's the only one that connects directly to the alpha amino group. Look what happens. Chaining together the alpha carbon and the alpha amine with an R group restricts the ability of the bond between the alpha carbon and the alpha amine to rotate. Not surprisingly, regions of a protein containing proline are less flexible. Methionine's chemical oddity is that it is one of two amino acids that contain sulfur. Sulfur's electronegativity is similar enough to carbon that there is fairly even sharing of electrons, meaning that sulfur bonded to two carbons in methionine is nonpolar, and the amino acid is hydrophobic. Methionine is noteworthy because the cellular machinery for making proteins uses it as the first amino acid for making almost every protein on Earth. So guessing the first amino acid of any protein is a bar bet you're likely to win. The first amino acid seen at the start of each protein is always a methionine. A subgroup of the hydrophobic amino acids is the so-called aromatic amino acids, named for the large and quite stable aromatic ring structures in their side chains. Phenylalanine is exactly what it sounds like, a phenyl ring of 6 carbons attached to an alanine. The R group is hydrophobic. If an OH hydroxyl is attached to the far side of the phenyl ring of phenylalanine, then we've made tyrosine, which is just a hydroxyl-containing version of phenylalanine. It exhibits some properties of hydrophilic amino acids, but the ring also makes it hydrophobic, so it can kind of go both ways. While phenylalanine is needed in the diet, people with a disorder, phenylketonuria, must limit their intake of it. And this is because they are unable to convert it into tyrosine, leading to an accumulation of toxic levels of phenylalanine and unusual molecules derived from it. Untreated phenylketonuria leads to neurological problems, seizures, and learning disabilities. Fortunately, newborn babies are now routinely checked for the disorder, and those that have it are prescribed a low phenylalanine diet that allows them to lead a more or less normal life. The artificial sweetener NutraSweet is a big no-no for sufferers of phenylketonuria, since it is derived from aspartame, which contains phenylalanine and aspartic acid joined together. Our final and largest hydrophobic amino acid is tryptophan, which has an R group of 9 carbons and 1 nitrogen in a structure known as an indole ring. Tryptophan is a starting point for the synthesis of many important biomolecules, including the vitamin niacin and the plant hormones known as auxins. Tryptophan is also a component of the neurotransmitter serotonin, which converts into the sleep-related hormone melatonin. High-dose supplements of pure tryptophan are sometimes marketed as a sleeping aid. These facts, however, also stoke an urban legend that tryptophan in Thanksgiving turkey is why we get sleepy after that annual binge. Well, if tryptophan were the cause, you'd be just as likely to fall asleep after a hamburger, since beef has as much or more tryptophan than turkey. Tryptophan can also be converted into a plant hormone, so maybe that's why you just veg out. Okay. So far, we've looked at amino acids with nonpolar, that is, hydrophobic R groups, which try to hide from water. When proteins fold, these are the amino acids trying to hide inside the folded molecule. Let's move on now to the polar amino acids. The first of these is cysteine, which has sulfur, but unlike methionine, cysteine sulfur is in the form of an SH group known as a sulfhydryl. The SH group of cysteine is reactive because the H+ proton can readily be lost near a physiological pH of 7.4. And loss of a proton leaves the side chain with an S that is negatively charged. The electrons in the sulfurs are lost in the reaction that creates the disulfide bond between them. It is not at all uncommon for a protein to have multiple cysteines. If the protein arranges itself so that the two cysteines are brought into close proximity of each other, a disulfide bond could form that is very strong and provides structural support to the protein. You can see the work of disulfide bonds in hair. Hair is made up of protein strands whose structure is stabilized by disulfide bonds. Chemicals used for straightening hair or for curling straight hair break these bonds and then remake them while holding the hair in the desired form. Those foul sulfurous odors when hair is being chemically straightened or curled, that's disulfide bonds of cysteine being broken and remade within the hair. Similar to the SH group of cysteine are the OH hydroxyl group in serine, threonine, and tyrosine. Unlike the SH sulfhydryl group on cysteines, the hydrogen on the OH of these amino acids does not come off readily at physiological pH. Unlike the OH groups found in carboxyl COOH groups, the OH groups of these R groups normally will not form ions. Instead, the OH stays together, remaining uncharged and happily hydrogen bonding with water. The simplest of these water-friendly amino acids is serine, which has only a single carbon in its R chain, just like alanine, plus an OH at the end. Serine plays a distinctive role as a signaling molecule in the brain, thanks to D-serine, another rare D-amino acid in our bodies. Meanwhile, ordinary L-serine is important for the synthesis of numerous cellular molecules, including proteins, membrane components, and nucleotides for making DNA and RNA. Threonine is similar to serine in many respects, and both function in the active sites of many enzymes. The largest hydroxyl-containing amino acid, tyrosine, is the starting point for the synthesis of many biologically important molecules, such as thyroid hormones, adrenaline, and also the nerve signaling molecule dopamine made in the reward circuits of our brains. All three of the hydroxyl amino acids in proteins can have their hydroxyl groups modified. Enzymes called kinases catalyze replacement of the hydrogen on hydroxyl groups with a phosphate, a modification hugely important for protein regulation. Phosphorylation can act as a sort of switch where toggling between the phosphorylated and unphosphorylated state determines whether a protein is active or not. The most common targets for phosphorylation are the hydroxyls of serine and threonine, but tyrosine phosphorylation is also important in cells for signal relaying. At physiological pH, two of the amino acids are notable for being acidic, meaning their R groups lose a proton, while three of the amino acids are basic, meaning their R groups gain a proton. All these amino acids have charged R groups at the pH found in cells. The acidic amino acids contain carboxyls in their R groups are aspartic acid and glutamic acid. Ionized forms of these are called aspartate and glutamate. Glutamate famously affects what we taste. Our tongues have receptors sensitive to free glutamate, which is responsible for the savory or umami taste of some cheeses and soy sauce. The sodium salt of glutamate, known as monosodium glutamate, or MSG, is often used for enhancing this flavor. The pKa values of the R group carboxyls of acidic amino acids are around 4, which means their protons are gone at the physiological pH of 7.4. They leave a negatively charged carboxyl group. The R groups of these two amino acids are identical, except that the aspartate chain is smaller, while the glutamate chain has one extra CH2 group. Glutamic acid gets its name from being discovered in gluten, the wheat protein. Aspartic acid was first obtained from a related amino acid called asparagine, which was in asparagus, hence the names. Both aspartate and glutamate help cells use protein as an energy source when supplies of sugar run low, or when a person goes on a low-carb diet. Aspartate and glutamate are important in managing ammonia in cells as well. Ammonia, or NH3, is a toxic byproduct of metabolism, and aspartate is a component in the urea cycle that helps to remove ammonia from the body. Glutamate, on the other hand, is extremely useful for its ability to accept or donate a molecule of toxic ammonia in reactions known as transaminations that pass amines between molecules. In the brain, glutamate binds to a receptor protein on nerve cells responsible for memory and learning. The receptor is also the site where psychotropic drugs such as PCP act by blocking glutamate's action on the receptor. Glutamate combined with ammonia makes a closely related amino acid called glutamine. The free ammonia becomes an NH2 amine, hence the name glutamine. The ability of glutamate to accept ammonia in this way is important for removing toxic ammonia from the brain, because glutamine is a rare amino acid that can cross the blood-brain barrier. In the same way, aspartate can swap out the OH hydroxyl and add an amine to become asparagine. Both asparagine and glutamine are uncharged amino acids, where the amine attached to the C double bond O creates a slightly polar group we call a carboxamide. This chemical change also means the R groups of asparagine and glutamine do not ionize at physiological pH, and this is why their R groups do not become charged, unlike their cousins, aspartate and glutamate. Interestingly, studies in 2018 have shown that lowering asparagine levels in mice reduced metastatic spread of breast cancer, a finding that could be very valuable if shown to work in humans. Glutamine is also approved for treating sickle cell disease. It is thought to work by reducing oxidative stress in sickled blood cells, restoring their flexibility and improving blood flow. At a cost of around $1 a day, glutamine treatment is 1/100th the price of other drugs, making it affordable to millions of sickle cell disease sufferers. The three basic amino acids, lysine, histidine, and arginine, are called basic because their R groups accept protons at physiological pH, giving them positively charged R groups. Lysine's R group is a long, flexible chain with a single positively charged nitrogen in the amine at the end. Animals in feedlots get lysine supplements to improve yields, especially if corn is their primary energy source, since most corn has relatively little lysine. Vegetarians who don't mix grains with beans, which are high in lysine, may encounter a similar deficiency. Adding lysine to grain products may eventually become obsolete with the development of high lysine corn through genetic research. Some people take lysine supplements based on unverified claims that lysine prevents cold sores or that it favors the release of human growth hormone, but there is little evidence of benefit for either claim. Similar but slightly longer is arginine with 3 nitrogens near the end of its side chain. Arginine plays an important role in the urea cycle as the source of urea. Arginine is also a source of a cellular signaling molecule called nitric oxide. Nitric oxide stimulates relaxing of blood vessel walls and improves blood flow. It is reported that arginine supplements may slightly reduce blood pressure. While considered generally safe in small amounts, arginine supplements are not without risk. They can worsen asthma and trigger the reactivation of dormant cold sore viruses and genital herpes. Large doses of arginine also have undesirable interactions with a variety of drugs, including blood thinners and heart medications. In general, large doses of any one amino acid may affect the metabolism of other amino acids, so beware. Remember that awful feeling during a cold and the accompanying congestion? Well, the inflammation there is caused by histamine, which helps mediate the itch response. You've probably popped a few antihistamines like Benadryl to avoid histamine synthesis from our next basic amino acid, histidine. Histidine is important in many enzymes and in the blood proteins myoglobin and hemoglobin. It is also the most chemically interesting of the basic charged amino acids. Histidine's ring contains 2 nitrogen atoms that share a proton when the pH is sufficiently low, giving the group as a whole its positive charge. Histidine's ring-shaped R group is the basic amino acid with the lowest pKa of 6, meaning at physiological pH of 7.4, the proton's gone. Losing a proton means histidine's ring is mostly uncharged because when a basic group loses a proton, the charge goes from +1 to 0. But reductions in pH can drastically increase the percentage of histidine rings with the proton on. Remember that the pKa of an ionizable group is the balancing point, where half of the molecules have the proton on and half have it off. Above that pKa, more molecules will have the proton off than on, as is the case for histidine here. Lowering the pH towards the pKa increases the percentage of molecules with the proton on. Now, pH changes down to the range of 6 or so are not feasible in cells as a whole, but such changes can happen easily inside of a protein, and that's where histidine works its magic. Their histidine's ability to flip charge from 0 to +1 or vice versa can be very beneficial in catalyzing enzymatic reactions. We've almost reached the end of our speed dating session with the amino acids, so let's finish with a bit of nutritional information. The U.S. recommended daily allowance, or the RDA, for dietary protein is about 50 to 60 grams for adults. So what about the amino acids? 9 of the 20 amino acids that make up our proteins cannot be made by our own cells. So those are called essential amino acids, meaning they must be in our diets. Plants and bacteria, by contrast, can make them all. So by eating them or animals that eat them, we readily meet our amino acid needs. Overall, it's rare, except for specific medical conditions or under very restrictive diets, to be deficient in an amino acid. 7 amino acids that are sometimes called semi-essential or conditionally essential recognize that children or other subpopulations may have difficulty synthesizing them in sufficient quantities. Speaking loosely, it's the bigger amino acids that turn out to be essential for humans, and the smaller ones our bodies can usually make. Tryptophan with 9 R-group carbons is essential, and so is phenylalanine with 7 R-group carbons. However, our bodies can, via a sort of Rubik's Cube path through a few intermediate steps, eventually add a hydroxyl group to phenylalanine and form the non-essential tyrosine, otherwise identical with 7 carbons. In fact, going the other way is possible too, and about 50% of our needed phenylalanine can actually be synthesized in our bodies from the non-essential tyrosine. And that's a more important point to take away. There is typically more than one pathway for getting from one amino acid to another. What that also means is that trying to supplement a single amino acid is more likely to have unexpected consequences than to deliver the targeted effects that may have been advertised or promoted. After all, what amino acids especially do is join with other amino acids. Put together a few, and that's how you get a peptide. Glutathione is an especially simple but important one made with just 3 amino acids: glycine, L-cysteine, and L-glutamic acid. Glutathione helps protect cellular structures from oxidative and heavy metal damage. And no, we're not talking about the effects of AC/DC or other rock group music on your ears. Many important hormones and neurotransmitters make use of very short peptide molecules. So let's wrap this up. Amino acids are fascinating on their own, but they become especially powerful as building blocks of proteins. This can be a good lecture to watch again after one or more of the other lectures to come. For now, let's conclude with something completely different. To review what you've learned, let's end with a song about amino acids. Now, some of these will have only 3-letter abbreviations to make it fit in the song, so see how many you can figure out. Lysine, arginine, and his, basic ones you should not miss. Allelu, val, and met, phylly, aliphatic set. Proline, benz, and cis has S. Glycine's R is the smallest. Then there's terp and tyre and phe, structured aromatically. Asp and glue side chains of R, say to protons au revoir. Glutamine, asparagine, bare carboxymitamines. Threonine and tiny ser have hydroxyl groups to share. These 20 amino A's can combine a zillion ways.